Thanks for coming tonight. I really appreciate this opportunity to share with you some of the research that my laboratory is working on right now. But before we get into talking about the science, I want to have a little audience participation. So, and this will be a show of hands if you don't mind. How many people like crossword puzzles or other kinds of word puzzles? And a few hands. What about jigsaw puzzles? That's one of my personal favorites. Okay. Another one of my personal favorites are logic puzzles. I see lots of hands going up with the Sudoku puzzles. What about 20 questions or other kind of guessing games? Yeah. Anybody like mazes or labyrinth type puzzles? More hands going up. My 14-year-old son's personal favorite video game type puzzles like Tetris. Anybody like mechanical puzzles like Rubik's Cubes? Okay. And the last kind of puzzle is a puzzle that's made up of lots of puzzles called meta puzzles. I don't know if anybody's experienced meta puzzles. Some of you might participate in things like the robotics competitions. Oftentimes they're meta puzzles where you do lots of different kinds of puzzles to make one big puzzle. What's shown up here is the MIT Mystery Hunt for 2009. This is a series, it's a meta puzzle they do every year where teams get together for a weekend and solve puzzles to come up with the ultimate puzzle, which is where this little coin is. And the team that solves it all these puzzles the fastest figures out where the coin is. But what all these puzzles have in common is the fact that they all revolve around a question that's difficult to answer, or they take a lot of patient thought and ingenuity to solve. Okay? And that's what scientists are. If you ask us, we pretty much all like to do puzzles because we like to spend time thinking about things that are difficult to answer, questions that are difficult to answer. And so for tonight, I'm going to be telling you about a meta puzzle. Or actually, I'm going to be telling you about two puzzles within a meta puzzle. A meta puzzle being a puzzle that has lots of puzzles in it that make up the whole meta puzzle. So what's the meta puzzle that I'm going to be talking about tonight? Well, it's the tobacco's effect on human health. Okay? Well, how big is the meta puzzle that we're talking about when we talk about tobacco's effect on human health? Well, to give you a sense of just how big a puzzle this is, 61.7 million people in the U.S. right now smoke. Okay? That's about 20% of the U.S. population. Just to put that in context for us here in Alaska, 160,000 people smoke here in the state of Alaska right now. That represents about 23% of our state's population, which means that Alaska is the second highest state for the percentage of population that smokes in the country. The only one that's higher is the state of Kentucky, which happens to be a state that produces a lot of tobacco. Okay? So it's not only a huge national problem, but it's a huge issue in the state of Alaska. Okay? So I said we were talking about meta puzzles. So the first puzzle is why does that 61.7 million people start smoking in the first place? We've all heard about the addictive effects of nicotine. Well, what is it before they actually get addicted to the nicotine that gets them started smoking? Okay? That's one of the puzzles that people across the country, scientists across the country, are working on to address the tobacco's effect on human health. Okay? Well, of those 61.7 million people, half are going to die because they smoked. Okay? So the second puzzle is is, which half of the 61.7 million people are going to die? Think about it, you might have seen walking in, people standing in a circle outside the door smoking. Why is person A smoking and person B standing next to them smoking? But person A is going to die because they smoked, and person B isn't. What is it about the difference between those two individuals that means one is going to live and die of something unrelated to smoking, and the other person is going to die because they smoked? Is it the way they smoke, what they smoke, their genes they express, their genetic makeup? That's one of the puzzles that people are working on to try and understand tobacco's effect on human health. Just to tell you about this image here, this is from an art exhibit that's traveling around the country right now. The artist is Chris Jordan. You see the name of the traveling exhibit called Running the Numbers, an American Self-Portrait. This artist creates works of art using the things that make up our society, issues addressing our society. This skeleton, or smoking skeleton, is actually made up of tobacco packs. There are 220,000 tobacco packs to make up this skeleton. That's half of the number of people who die every year from smoking in the U.S. Okay? Kind of an interesting way of thinking about numbers that affect our society. Well, what's the third puzzle? So of those half of the 61.7 million people that are going to die, what are they dying from? Well, are they dying from lung cancer? That kills about 130,000 people a year. Or maybe they're going to die from heart disease. That kills another almost 130,000 people a year. From smoking. These are smoking-related deaths. Or just under 100,000 people die from chronic obstructive pulmonary disease. These are things like emphysema and chronic bronchitis because they smoked. Or perhaps from one of the other myriad causes associated with smoking, things like bladder cancer or smoking-associated strokes. Okay? So that's another puzzle that we have to figure out or understand if we're thinking about how tobacco affects human health. Well, the question is, how does tobacco contribute to each of these diseases? Well, that's the third, the fourth, the fifth, and so on puzzles. Okay? Starting to sound pretty daunting tasks to try and understand the effects of tobacco on human health. Well, this is why, as a cellular immunologist and a biomedical researcher, I belong to a group. Biomedical researchers tend to be what are called reductionists. Well, what are reductionists? Well, reductionists attempt to understand very complex systems by breaking it down into smaller, simpler pieces. So if you look at this, graphic of a jigsaw puzzle, you think about a group of people sitting around the table working on a jigsaw puzzle together as a team. Maybe one person focuses on the sky, another person, my personal strategy for addressing jigsaw puzzles is the first thing I like to do is find all the edge pieces to see where the perimeter of the puzzle is. Okay? Another person, like my 4-year-old daughter, she goes for the brightest colored pieces and starts working on putting those together. Or my husband's strategy is he tends to focus right in the middle, smack dab middle of the puzzle. Okay, kind of builds out from there. But in any case, it's a team of people working together in their own kind of area, personal preference, if you will, trying to work on their piece of the puzzle, and then putting all of that knowledge together to understand a much more complex puzzle. Okay? So working as a team, and breaking it down into smaller pieces to understand a much more complex question, like tobacco's effect on human health. So tonight I want to talk about 2 of those puzzles that are part of this really large meta puzzle, the tobacco effects on human health. So the first puzzle that I want to talk about is the effect on lung architecture. Well, hopefully, as you're sitting there, if we were to dissect out your lung, this is what it would look like. This is what a nice, healthy lung would look like, or looks like. This is a picture from the American Lung Association. And you'll notice that it has a nice, smooth surface, a very defined edge to it. It looks pretty clean. From my perspective as a biomedical researcher, it's kind of pretty, okay? But if you have lung cancer, and we were to dissect out part of your lung, it might look like this, okay? You'll notice it's very brown. It doesn't have a nice smooth surface. It doesn't have clean edges. Here's the tumor up here, in fact. You see that that's pretty much changed the architecture of the nice, clean lung. Well, maybe you don't have lung cancer, but you have emphysema. And if you have emphysema, your lung might look like this. There's no smooth surface at all. In fact, there are all these bulbous projections of tissue. Think about taking a balloon, blowing it up, and then squeezing, pinching off part of the balloon, and it kind of puckers out and looks very thin. Well, that's kind of what this is like. And if we took a section of this lung, very thin section, you see it here. This area down here is about what the normal lung would look like, very densely packed with lots of cells. But if you look at the section, the portion of the section up here, there's lots of holes. It's missing a lot of cells. All of that lung architecture is now gone in a person with emphysema. This is why people with emphysema have a great deal of difficulty breathing. In fact, I think there was a hands-on activity with a licorice stick over there earlier this evening. Some of you might have tried to demonstrate what it's like to try and breathe if you have emphysema. Okay? So you lose the architecture of the lung, whether you have lung cancer, whether you have emphysema. So how do we get from this nice, healthy lung to this, or to this? Well, to understand that question, or to address that question, we need to start at the beginning of how lungs are formed in the first place. So this is a diagram from a group at Stanford University. In fact, this is pretty new data. This was published just a year and a half ago. So really new. Before that, we really didn't exactly understand how the lung formed. Well, what this group at Stanford University discovered is that just three geometric processes could account for the development of the entire structure of the lung. Okay? And they're nicely color-coded here. These are pseudocolored images of embryonic mouse lung. So E stands for embryonic. The number for each picture represents the day after fertilization. So at 12 days after fertilization in the mouse, you start seeing the very primordial lung developing, and it continues to develop throughout the process of development in the mouse embryo. Well, these blue colors here represent the basic fundamental structure of the lung, the major branches. And that's called domain branching. So every once in a while, the cells, as they start growing to form the lung, simply branch out at a 90-degree angle from the main column, if you will. Okay? And that sets up the overall structure to the lung. If you look at how the edges of the lung are formed, that's shown in the green here, you get what's called planar branching. Well, to understand what planar branching is, if you were to take your hand and you look at it, it creates a nice flat plane, right? Well, planar branching is simply, and this is going to date me, because this is going to make me look like I'm really into Star Trek, and I'm not that big of a Trekkie. But planar branching basically creates the Vulcan hand symbol, okay? It separates out cells from one another, but it stays within the same plane, okay? The third type of branching that creates the bulk of the lung, that makes up all the interior of the lung, as shown here in red, is orthogonal branching. And what we mean by orthogonal branching is we take our hand again and we've got our plane. Well, instead of branching in the plane of the hand, we branch out of the plane. So we create different planes. Okay, that's what orthogonal branching. So just those three processes can account for the entire development of the lung. Okay? And what this looks like in action is shown here. Okay, this is from Zena Werg's group at the University of California, San Francisco. And she studies branching morphogenesis in mammary tissue. But as you can see, there are structures that are in the plane of focus, but there are other structures coming out of the plane of focus. Okay, so that's that orthogonal branching. And in the end, you get a structure that looks very much like what the lung looks like if you dissect down to smaller and smaller structures within the lung. Okay? Kind of looks like a grape cluster. Okay? So that's what branching morphogenesis is, and that's how the lung forms. Okay? Well, surprisingly, although that really elegant experiment was done to look at the structure, we really don't know too much about what signals are actually controlling that process. What's shown here is really about what we know at this point in time. And you notice it kind of looks like a teeter-totter. Well, what we do know is that it's a balance between two main signals, one defined as FGF, and one from SPRY2. Well, what are these? And you'll see some more minor players, other factors that influence these two major factors. Well, it's a balance between FGF, which stands for fibroblast growth factor, and SPRY, which stands for sprouting. Okay? FGF comes from the connective tissue that the primordial lung is going to develop in. Okay? That connective tissue produces FGF. And that tells the primordial lung epithelial cells that it's time to form a lung. And you'll notice that you see a little straight arrow here. Well, FGF tells the cells to grow, proliferate, and to grow out in a straight line, if you will, to make straight tubes. Okay? Well, as FGF is telling those cells to grow and divide, to make straight tubes, it also tells the cell to start making sprouting. And so as the balance between FGF and sprouting changes, the sprouting expression increases, the epithelial cells sense that increase in sprouting and start branching. And so as the balance switches back and forth on the teeter-totter between sprouting and FGF, the cells in the lung either grow straight or they form a branch. And it continues on in that cyclical fashion until the lung is formed. Well, why do we care about these genes? Or how the lung develops in the first place? Well, the same kinds of genes, and perhaps even these very genes that are involved in developing the lung, are also involved in helping the lung repair when it gets damaged. Okay? We think the same kinds of processes might be at work to help repair damage that might have been caused by smoking. So we posed the question of which genes might show altered expression when the lungs are exposed to smoke. we can't very well ask people to go in a room and smoke for us and then give us their lungs. It's just not very ethical. So how do we attack that problem, that puzzle? Well, we come up with what's called a model system. And for this particular set of experiments, we used a model mouse. A mouse that can develop emphysema. So how do you smoke a mouse? Well, you get a big room and you get a big house to keep the mouse in. Okay. You also get a very large machine to smoke lots of cigarettes for you. This is a setup at my laboratory when I was at Lagos Respiratory Research Institute in Albuquerque, New Mexico. They have lots of rooms like this for studying things like tobacco and diesel exhaust from diesel engines and gasoline engines and wood-fired furnaces. But in this particular setup, this machine smokes the cigarettes for us. And it smokes it just like a human smokes it. And then we can collect the smoke and pump it into the housing chambers, these big stainless steel rooms where the mice live. So if you do that for 6 hours a day, 5 days a week, for about 4 months, the mouse develops emphysema. Pretty much just like the emphysema that we see in a human being. Right? So what you're seeing here, this is a healthy lung, a section, a very thin section, from the healthy lung of a mouse that was just exposed to nice, clean, fresh air. And here's the section from the mouse that was exposed to smoke, as I described. And what you see, just like the human lung section I showed you earlier, is this mouse develops large holes in his lung, or her lung. I'm not sure if it was a male or female. So the architecture in the animal is completely changed when they're exposed to the smoke, just like a smoker, a human smoker, is exposed. Right? They develop large holes, they lose the cells within their lungs, and they have trouble breathing. Okay. So I said, well, we were interested in knowing whether gene expression was changed within these animals. Perhaps like a human might be altered if they were exposed to smoke. And so we wanted to look at the genes and how they were expressed. And to do that, we use a technology called microarrays. What microarrays allow us to do is look at lots of genes simultaneously. What you're looking at here is actually a class slide, much like you might use in your middle school or high school science class. Okay? And what each one of these little dots is DNA from a gene that represents a gene in the mouse. This represents about half of the genes in the mouse. This is about 7,000 different genes. Okay? And you'll notice that some of the spots are bright and some of the spots are dim. And it looks like they might have different shades of color. Well, we're comparing the gene expression from a mouse that got fresh air versus a mouse that got the smoke at the end of that 4 months of sitting there smoking like a person smokes. And what we found is when we looked at all the genes, one of the things that popped out was the fact that a number of the genes that were altered belong to pathways that are involved in lung development. So if you remember back to our teeter-totter here, and the group of signals that cause the cells to proliferate and grow in straight tubes versus the signals that cause the cells to divide and to create branches, you'll notice that in the emphysema model, we found that genes that influence that straight growth were altered, the wingless or Wnt genes. Genes from transforming growth factor beta and bone morphogenic pathway. This is a large group of genes that influence the branching process, were altered, as was the gene family of sonic, or the pathway called sonic hedgehog. And this is another pathway that influences that branching. It sort of inhibits the outgrowth and favors the branching process. So the first part of the puzzle that I've shown you tonight is that smoking alters the expression of genes involved in lung development, and we believe those genes are involved in repair as well. And these are altered in a mouse model of emphysema. So, being exposed to smoke alters the expression of genes that might influence the capacity of the lung to be able to fix itself when it gets that damage from the smoke. Okay? So that's the first puzzle that I wanted to tell you about tonight. Well, that's all well and good. We have more information about emphysema now than we did before we went after this question. But there's two issues to it that make it less than maybe perfect. One, emphysema is at the very end stage of disease. There's almost nothing we can do at the point that we sample these animals to fix the problem. Just like the picture of the lung from somebody with emphysema was at the end stage. The damage was so severe. There was really the best developmental team in pharmaceuticals or other kinds of therapeutics would be hard-pressed to come up with a strategy to fix the problem. So we really need to understand what's happened when the person's first exposed to the smoke, not at the end of all of the damage. So that's the first thing that we need to go after, is trying to do it earlier, address earlier problems. Second thing is the fact that it was a mouse model. You know, I'm certainly, as a biomedical researcher, not in the business of fixing mice. My goal is to understand human health. We learn a lot from models, but ultimately we have to understand what's happening in humans and human cells. And so that's what I want to talk about in the second puzzle for tonight, is breaking the barrier within human lung cells. When I showed this video of the branching morphogenesis, I think everybody can agree. It wasn't as if one cell grew over here, and another cell grew over there, and another cell grew over there. They grew next to each other. They grew out from this starting point here. These few small cells are connected to one another. Well, that's because lung epithelial cells, like all epithelial cells in our bodies, want to stick to one another. That's their job, that's the way they like to exist, is stuck to one another. Well, how do they stick together? Well, they stick together through what's called a junctional complex. Here's one cell, here's another cell. This is actually an image taken from the cell biology textbook that we use down at UAA, cell and molecular biology. And you notice that the junction is made up of three things. TJ stands for tight junction, AJ stands for adherence junction, and D stands for desmosome. Now I think you can notice that it looks like there's space between the two cells at the desmosome and the adherence junction. But if you look at the tight junction, it looks like the two cell membranes are right smack dab up to each other. And if you look at this from above, you'll notice that the tight junction forms a belt all the way around the cells, between each adjacent cell. the tight junction creates the barrier that separates the outside world from the inside world. The inside of you. Well, why do we care about being separated from the outside world and the inside world? Well, if you look at this picture, this is from San Francisco. These are so-called painted ladies. And imagine there's a couple people standing down here. Hopefully they're not in the audience and I don't offend them. But let's just imagine they're burglars. And they have keys to every one of these houses. Except the key only fits the back door. Okay? And let's imagine that there's a fence with a gate in between each one of these houses. Well, which houses do you think the burglars are going to go and hit with their key to the back door? The house with the intact fence with a locked gate? Or the house whose fence is falling down and the gate is wide open saying, come on in? Okay? That's why we care about the barrier, because the tight junction create a fence and a locked gate between adjacent cells, just like, and help to keep out the burglars, just like fences and gates do in our own homes. And in this particular instance, what we mean by burglars are things like viruses, some bacteria, and even some of the more chemicals in tobacco smoke. There are 4,000 chemicals in tobacco smoke, about 60 of which are known carcinogens, and a whole host of other kinds of chemicals that we know to be harmful. Okay. So the tight junction creates a barrier to keep those kinds of things in the outside world and prevent them from entering the inside world, the inside of you. That's one thing the tight junctions do. The other thing they do is that they create what we term a polarized cell. Just like the house in the picture before has a front door and a back door and a side, maybe a side. Well, the tight junction creates the top or the front of the cell compared to the back or the bottom of the cell. If you take away those tight junctions, you take away the top or the apical surface of the cell. And instead of having separated lipids, different lipids at the top of the cell, from the sides and the bottom of the cell, or different proteins in the side and the bottom versus the top of the cell, if you take those tight junctions away, everything gets mixed up. You lose the architecture of the cell. And all of the functions of the cell that are dependent on being separated from one another, or having certain kinds of lipid molecules in the membrane in a certain area, are lost as well. So that's why tight junctions are such a critical component to the cells that are in the lung. Okay. So just like I said when it came to the animal line, I said we can't put a person in a room and have them smoke and then ask them to give us their cells. Not ethical. So we had to come up with a strategy for being able to still study. We wanted to study human cells, but we needed to be able to do it in the laboratory. And this is what we've come up with. This is an in vitro smoking machine. What it allows us to do, here's our cigarette down here. This is computer controlled. The rest of the computer is outside the image here. The cells are up in these glass and stainless steel chambers. But what this system allows us to do is to smoke the cigarette just like a person smokes it. The computer can have the pull air through the cigarette the same volume that a person does when they're smoking a cigarette for the same amount of time. and then inhale that smoke into the cells in our chamber. And then in the same amount of time that a person would, exhale the smoke. So what does this look like in action? Well, it's difficult to see real smoke because it's such a small amount that the company that built the chambers has developed a video for us so you can see. So our cells are sitting down here, and here comes this smoke. Okay? Down an aerosol inlet, just like the smoke will be coming into the lung of a person. Down their trachea, down their bronchi, into their lungs. And in the same kind of time frame that a person inhales the smoke and then begins to exhale the smoke moves in and out of our system. And this goes on in a repetitive fashion for about 8 minutes, because that's how long a person, on average, takes to smoke their cigarette. And you can see that repetitive cycling appear on the computer screen. So if we do this with cigarettes, and this just takes one cigarette. Today, when I'm going to show you just one cigarette. And we ask what happens to that barrier function of the tight junctions when we smoke a cigarette. One of the measures of that barrier function is that it prevents ions from flowing between the adjacent cells. So it works like a resistor, almost like a resistor in electronics. Okay. So we can actually measure whether it's functioning to prevent the flow of electricity. And that's what this little instrument does here. It looks like little chopsticks. And we can send a pulse across the cells and ask, is the pulse stopped or does it go through? And so what we're looking at here is the effect of smoking either a high tar, a medium tar, or a low tar cigarette. So what's termed a full flavored cigarette, a light cigarette, or an ultralight cigarette. And a loss, what we're looking at is a percent change, so a loss in that resistance, meaning the tight junctions aren't working. They're no longer preventing the flow of electricity. And as you can see, it doesn't matter what kind of cigarette you smoke. Just smoking one cigarette causes the tight junction function to be lost. Now, over time, that function starts to recover. Interestingly, if you smoke the full flavor, that high tar cigarette, it actually recovers back to exact 100%. The tight junction completely reforms as if nothing happened to it. But if you look at the light and the ultralight cigarettes, we never see the tight junction function completely restored. And this is one of those ones where, at this point, we don't know the answer to why this happens. We're still working on it. But it very clearly, repeatedly happens that the light and the ultralight are worse than the full flavor. So anybody who says that light or ultralight cigarettes are less harmful is selling a bill of goods. They just don't know what they're talking about. So those are ions flowing between those cells because the tight junction is not working. Well, ions are great, but they're just little tiny single element molecules. In biological systems, we're usually interested in very large molecules, things like proteins, or even larger entities like organisms, like bacteria or a virus. So we wanted to know whether large molecules could get through as well when we smoke the cigarette. And that's what we're showing here. This is a fluorescence microscopy image. And just to let you know what the colors are here, the blue fluorescence is a dye that binds to DNA. So this is showing you where the nucleus of the cell is located. The green marker is showing you where one of the proteins that make up the tight junction is located. It doesn't tell you whether the junction is working, it just tells you that one of the proteins that make the junction up is present. And then what the red dye is showing is a tracker molecule, serum albumin. Serum albumin is the most abundant protein in your blood. Okay? And in this particular experiment, we're using it as a tracker. Okay? And we've attached a red tracking fluorescent dye to it. So we smoke the cells in that system like I showed you, and add the fluorescent tracker molecules. And ask, do we see it move from the top of the cell to the bottom of the cell, between the adjacent epithelial cells? And when you actually look at this in the microscope and look at the red dots, you see there are more red dots in the bottom portion of the cell, between the cells, between the adjacent cells, than are on the top. And we can actually sit here and count the red fluorescence and quantitate. And that's what's shown here. So as you increase the amount of smoke that the cells are exposed to, you increase the amount of red tracker molecule that is located between the epithelial cells, indicating that the tight junction function's been affected again by the smoke exposure. Okay? If we look at the air, you'll see there's no or minimal red tracker molecule located between adjacent epithelial cells, because in the air condition, the tight junctions are working properly. So what does the tight junction actually look like? Well, this is a cartoon version of what the junction looks like. It's made up of two proteins, or actually three proteins, excuse me. Two of them, claudin and occludin, actually project out through the cell membrane to pull the two cells together and interlace, much like your fingers can interlace with one another. Okay? And you'll notice that it's not just a single interaction, but there are multiple interactions making up that tight junction. Because as the term implies, the connection is very tight. Okay? And as we saw in the electron micrograph at the beginning of this section of the talk, the two membranes are very close together. Okay? Well, these proteins are connected to protein in the cytoplasm of the epithelium, the long epithelial cell. And it's actually a family of proteins called zonal occludins. 1, 2, and 3. Not very particularly fancy names, but those are the names nonetheless. And what those proteins do is it links the tight junction to the cytoskeleton of the cell. And the cytoskeleton is made up of actin and myosin, just like the actin and myosin in your muscles that make your muscles contract. Well, why do I bring up contracting muscles? Well, one of our hypotheses is that the actin and myosin actually pull the junctions apart in the cells exposed to smoke. And so cause these to open and close depending on whether their cells are in a healthy state or a smoke exposed state. Much like the Panama Canal works. So if you think about a canal with locks, there are train tracks that run along the locks in the Panama Canal that help move the ships through the locks and help them have the gates open and close. So just like the gates of the Panama Canal, those occludin and claudin proteins can open and close, to open and close the tight junctions. And so our hypothesis is that sometimes the smoke alters the cell cytoskeleton to open all of the gates at once. That allows that electrical current bridge. Okay? At other times, we see only one gate opening and closing, and then another gate opening and closing. And that's how it looks like the large molecules might be moving through. So we don't even have to have all the junctions broken down all at once. They might only partially have to be broken down, but that would still allow large things to get through. So where do we go from, So the summary for this part, or this puzzle, is that smoke from the most popular cigarettes, and I didn't tell you what brand, but Marlboros represent the largest fraction of the U.S. market, and those are the cigarette brands that we use in the laboratory. That's one of the cigarette brands we use. And the data I showed you tonight was for Marlboro brand cigarettes. But those alter the tight junction function in long epithelial cells exposed to cigarette smoke. And that was smoke from a single cigarette. So where are we going from here? Well, we're trying to understand the signals, the biochemical reactions within the cell that control the relationship of the cytoskeleton to the tight junctions. Because as I said, our hypothesis is that smoke changes the regulation, causing the cytoskeleton to actually pull the junctions apart, opening up the cell, or the space between the cells, creating no longer a barrier between the outside world and the inside world. So it's disrupting that barrier. And it's also disrupting the barrier that creates that polarized structure to the cell. These cells no longer have a top and a bottom. It's a mixed up mess. And so that's where we're going with this. Well, before I open it up for questions, I would be remiss if I did not acknowledge the people that work on this project. As I said, science is a team effort. I have my team, there are other teams, as I showed you some work from San Francisco, Loveless, in New Mexico. There are teams all over the country working on this giant meta puzzle. And this is my team right now for this year in the lab. One of the things I wanted to point out is that a number of these are in fact, many of them are undergraduate students doing research. Okay? Some of them, like Carolyn Jerome, are actually a high school student working in the laboratory. She's from Kodiak. And then other people that have worked on this project and other projects in my group at UAA, and then my group that was at Loveless, and in particular I want to acknowledge Dorian Oliveira, who was a graduate student who was the first to build the prototype in vitro smoking system, bringing all those different kinds of pieces of equipment together and making it work. And I would be remiss if I didn't acknowledge the people that pay for this. This is an example of our tax dollars at work. Pretty much from the time I first stepped in my foot in a laboratory, my work has been funded in one way or another by the National Institutes of Health. So that's an agency of the federal government with help whom we could not do this work. My home institution, UAA, Alaska Embry, the state of New Mexico, who used some of their tobacco settlement money to pay for the very expensive mouse exposures. Those are very expensive experiments to do. Loveless Respiratory Research Institute, who also funded that work. And I have to thank Johnson & Johnson, who gave a fellowship to Dorian so that he could do his PhD dissertation. And with that, I'd be happy to answer any questions you have. Thank you for your attention.